Hello, everyone. We are excited to have you here with us today. I'm Zhengxi, Marketing Director at VCLS. I will be your host, moderator for this session. Thank you for joining us. In today's session, we will explore how quality by design, when combined with an agile development principle, can help address some of the biggest challenges in pharmaceutical development. For example, development timelines, product robustness, and regulatory alignment. Please welcome Nicole Cohen, CMC Senior Director at VCLS. And Nicole has more than 30 years in pharmaceutical industry, and she is focusing on CMC regulatory in this particular field. And during her work in pharmaceutical industry, she has applied QbD to chemical and biological products at different stages of development, from early stage to late stage until commercialization. And please also welcome Thierry Bastogne. He is a professor at University of Lorraine in data science applied to pharmacology. Thierry has also founded a company called Cybernano, a digital biotech which has developed Easy QbD, a web platform aimed at democratizing the use of quality by design. In recent years, Thierry's research activity has focused on the areas of the use of AI to improve precision and accelerate QbD studies. Nicole and Thierry collaborated last year on an e-learning program for EIT Health EU commissioned, which will be delivered this year to pharmaceutical industry. Just briefly, some housekeeping notes. We will have 45 minutes presentation and 15 minutes Q&A session. So please use the question box to submit your questions. And as usual, the session will be recorded, and we will share the replay shortly after the live session, and we will be sending you a link where you can access the replay. And also, we will be running a few poll questions throughout the webinar so that we can get a better sense of your perspectives and experience. And we encourage you to participate. With that, let's get started. Here is the agenda of today's webinar. As we begin, we will start with a brief introduction to QbD and Agile QbD, followed by an overview of key regulatory guidelines and the FDA EMA joint pilot program. We will then move into a practical training case studies focusing on monoclonal antibody process to illustrate criticality assessment, experimental screening, the definition of operating regions, and validation of a design space. We will conclude with some key takeaway messages and a Q&A session at the end of this live session. I would encourage the audience to submit your questions throughout the webinar by using the Q&A function. We will address them, as many as possible, towards the end of the session. And before I pass the floor to Thierry and Nicole, I would like to launch the first poll question, just to get an idea of the audience experience. Where is your lead program today? You can pick up one answer. Or the result, it's I would say a mix of development stage. Many of them, the majority of the audience here, they are at early preclinical program. But you will see that this diversity is actually ideal for today's topic because QbD and specifically Agile quality by design can be really applied at different stage. And Nicole and Thierry will talk about more precisely about it. Now I hand over the floor to Thierry.
Thank you, Zhengxi. Good evening, everybody. So let's introduce quality by design. QbD is often defined as a systematic science and risk-based approach to product development. And the principle of QbD is to build quality into a product from the very first stage of development. But unfortunately, in practice, it is not really the case because QbD often starts after the phases to be clinical trials and not before. And in the same time, we observe that the main critical step of development is during the preclinical development because the risk to fail is about 90%. So this is the first motivation why we tried to simplify the QbD to be adapted to the preclinical development. And this modified and new version of QbD is named Agile QbD. And so now, before to present Agile QbD, the motivation in the next slide. And there are three main reasons to apply QbD. The first is to reduce time of development. We have now a lot of papers that estimate between 20 and 40% of reduction of time if QbD is well applied. And another and second reason is to optimize the quality not only of the product, but about the manufacturing process. And so finally, QbD bring an added value to the product after the development. But also another aspect is not to forget that QbD was initiated by FDA. So of course now it's also a way to facilitate the approval of dossier by the different drug agencies. This is the last reason. And now we're gonna start to define what I mean by Agile QbD. In fact, even if most of the QbD ingredients are now relatively well understood, in fact, in practice, the overall approach to apply QbD still lacks of clarity and flexibility. So that's the reason why we decided to combine QbD with another well-known approach called the Scrum Agile, which is an approach very used to manage complex projects. And the idea was to associate the two advantages, the flexibility of management coming from the Agile approach with the scientific precision and risk analysis coming from the QbD. And if you wish to get more details about this hybrid approach, I let you the reference of the paper we wrote last year, and it is an open access, so you can get it freely. So now, before to go deeper in the Agile QbD approach, instead of using always the same acronyms on CQA, IPC, CMA, CPP, I prefer using words like output and input. And by output, I mean the critical quality attributes or in-process controls. That is all the properties of the product we want to control in order to meet some specification, for instance. And to drive the output, we need to modify freely or to change freely some inputs, and it corresponds to material attributes or process parameters. So I'm going to use inputs and outputs during all my presentation. And now we can go to a presentation of Agile QbD. And Agile QbD relies on a core concept, the concept of QbD sprints. And QbD sprint is composed of 3 ingredients. The first one is a development question. And I give an example of question below. For instance, what are the temperature and pH values that maximize the bioreactor yield? We're going to see another example in the case study. We will investigate a few slides later, sorry. But that's potentially a blocking question. And we have to solve that question to progress in terms of development. And it's really important to formulate clearly and simply the different questions of development. And to address the question, we implement a study cycle represented by the blue cycle, you can see. And I will present in detail that study cycle in the next slide. And at the end of the study cycle, so this is a scientific part, in fact, of the sprint. And at the end of this scientific study, we use the conclusion to take 3 potential decisions. That's the last part of the sprint. Potentially, the conclusion is positive. It means that we have solved the question and we can go to the next question. We can increment the development. Sometimes the conclusion is not complete. We need more accuracy, we need more data. So we, in this case, in this situation, we often iterate several times the cycle to design new experiments, to get data, and finally to get a clear conclusion. And sometimes, unfortunately, the conclusion is negative. So we demonstrate that we cannot solve the question. And it could be seen as a kind of alarm for the management team to say, okay, potentially we have to change the target product profile. We can change some ingredient, we can change the final indication or the mode of administration. And sometimes we can wonder if it would be better to give up the project to save money. So this is a different situation, but the architecture is always the same. So now I zoom on the sprint. So this study cycle is composed of 5 steps. The first step is named PPK. PPK for profile, process, and knowledge. Profile, it's something typical. That's the target product profile. So finally, a kind of profile of the product, but also the QTPP for the quality target product profile. So a list of quality attributes. But we have also to indicate the process because product and process goes together. So it's really important to also to decompose and to present the process for the manufacturing of the product. And the last aspect is more and more important. This is a knowledge management because you can imagine that sprint after sprint, we produce new data, new knowledge. And now with the emergence of artificial intelligence, it's really, really important to store, to organize the database to really make the knowledge very useful. And why? Because if you go now to the second step, called IOM for input-output modeling, this is a step in which we implement critical analysis to finally detect which inputs and which outputs are really critical. And this operation is done only based on the available knowledge. This is why the first step in terms of knowledge management is now more and more critical. But sometimes, unfortunately, the data available or the knowledge available is not enough. And so we have to design new experiments and to collect new data. And we are using some dedicated statistical methods to design experiments. And if the design is feasible, then we carry out the experiment during the step number 4. During the experiment, we collect data. And the data are analyzed in a statistical study during the last step, during the statistical analysis. And I have to stress that the steps 3 and 5, design of experiment and statistical analysis, have to be strictly suited to the type, the nature of question to be investigated. So it's really important for the success of the operation. And now, before to show you the first the study case to show how it works, I let Nicole to present you the regulatory aspect associated with the QbD.
Thank you, Thierry. So in the next few slides, I will present you the main guidelines related to quality by design, and it will be also the opportunity to remind some definitions that we mentioned before, on concept that we mentioned before. So those are ICH guidelines from EU, US, and Japan, which are the ICH countries, and I will also talk about a joint assessment pilot program on quality by design that occurred a few years ago, managed by FDA and EMA. So in this slide, I have summarized the main ICH guidelines related to quality by design, which were issued over the last 20 years, and that you should know when applying quality by design to your development. The first guideline issued is ICH Q8, a very important guideline, which initiates the main concepts of quality by design in regulatory dossier, with applied to the drug product first, with the notions of QTPP, quality target product profile, which is derived from the TPP, and also critical quality attributes of the drug product, which determine the quality expected of the product, the critical process parameters, and critical material attributes, which have to be determined in the manufacturing process of the drug product. The notion of design space is also described in this guideline, which represents the space portion where the process parameters, and especially the critical process parameters, can be variable without impacting the CQAs and then the quality of the product for the parameters studied anyway. And finally, the control strategy, which defines the level and where the controls should be performed all along the process of the product. In Q8, I mentioned risk analysis and risk assessment to be performed, and those tools are described in ICH Q9 guideline. Tools like FMEA, we will see later in the application later during the case study. ICH Q10 is related to pharmaceutical quality system and includes those notions essential for the continual improvement of the quality of the product during development, but also post-approval. And as Thierry mentioned, knowledge management is really the key in the pharmaceutical quality system to accumulate knowledge of the product. Then we have the guideline ICH Q11, which is parallel to ICH Q8 for drug product, but applied to drug substance. I jump to the recent guideline now, ICH Q14, which describe quality by design, but applied to the development of analytical procedures and descriptions of concepts of ATP, analytical target profile, which is the same as a QTPP for the drug product, and the notion of MODR, method operational design region, which is the counterpart of the design space for the analytical procedure in terms of robustness of analytical procedure, which is very important when analytical methods are developed. And finally, ICH Q12, which is the guideline for the life cycle management to follow up the quality of the product, especially during the CPV, continuous process verification, and all along the life cycle of the product, where the process will be followed during routine production and maintenance, checking for possible drift in CPPs or in the established conditions, if they are determined. You will see later in the case studies how those established conditions can be defined. Next slide, please. Thanks. On this development plan, you can see all those concepts we have just mentioned and when to generate them. You begin first in preclinical with a QTPP that is derived from the TPP, and all along the phase 1, 2, 3, and based on the knowledge, experimental data of the product and process, you will be able to determine the CQAs, the CPPs, the CMAs, important for the quality and the process of the product, until you arrive at on the design space notion, And all those key elements will be filed in your dossier for commercial and will be followed up during the lifecycle management and post-approval. As Thierry mentioned before, with the Agile QbD, you can begin quite early in phase 1 to determine those important concepts, rather than later on with, I would say, traditional QbD. For determining those concepts, the tools in the blue box below will allow you to perform risk analysis and FMEA that we will talk later on in the case study. In addition to ICH guidelines, I would like to talk briefly about the joint pilot program managed a few years ago by FDA and EMA on quality by design. The purpose of this joint pilot program was to enable a parallel assessment of quality by design related topics of the regulatory section of marketing authorization dossier submitted to both agencies, and the assessment of both agencies were more focused on QTPP, CQA, design space definition, and design space verification. This program was planned to last 3 years, but was extended to 5 years because many applications were submitted, 16 application requests, including CTT dossier, post-approval changes, scientific advices related to QbD were assessed by both agencies. The outcome of this program is really a very strong alignment in the expectations of regulatory aspects, especially on the items assessed on specifically design space, real-time release testing, and also, which notion very important, the control strategy. Thank you. From this pilot program, FDA and EMA have assessed the need to complete the ICH guideline I mentioned before, to give more details to pharmaceutical industry when submitted QbD in regulatory dossier, and issued in complement other documents, which are question and answers and lessons learned related to especially design space verification and the level of details to be given in the regulatory dossier. And at the end, the lessons learned confirmed a complete alignment in the expectation by both agencies. It has to be noted that the third ICH country, Japan, was an observer and participated in the question and answers issued and was also very well aligned with the expectations. Next slide, please. And so now what about future? As you know, probably a draft guideline, ICH M4Q second revision on the structure of the CTT dossier to be submitted for commercial product was issued last year. This draft guideline includes completely all the QbD concepts we have mentioned today, and they are completely integrated in Module 2.3 and Module 3, which are the CMC modules, as you know. So the QTPP has to be given, it's no more optional, with formal CQAs, CPPs, design space possibly in the Module 2.3, and all the risk analysis to support those determinations will be given in Module 3. A part is also given to analytical QbD, and a very large space is assigned to the overall control strategy, which is very key in this new format of Module 3 and 2.3. So you will find all the concepts we have already mentioned, and in this draft guideline, we really see that all the two modules, 2.3 and 3, are very interlinked, and progression all along the dossier from the QTPP until the control strategy. This has to be really emphasized in your CTD dossier. Also, this draft guideline emphasizes the applicability of QbD to biologics, ITMP, and complex products, much more than before, when it was rather more applied to small molecules. So you can see that the CTD becomes really a dynamic, knowledge-driven dossier including the life cycle management of the product, very structured, granular data, supporting ECTD 4.0 format, which is the new format, and of course supporting, including artificial intelligence and machine learning inside the dossier. And in silico, possibly if you're applying in silico. Next slide, please.
Thank you, Nicole, for the introduction of the relevant guidance, and Thierry for the introduction on what is QbD, what is Agile QbD. And before we move into the case study, I would like to introduce the next poll question. What is the biggest challenge you are facing while during developing stage? It can be multiple choices. I'll share the results. What stands out here is that many of these issues, they are actually quite closely connected. It's not just one challenges, but many of them. And I think that Thierry and Nicole will show you precisely in the case studies, and you will see how we can tackle them through the Agile QbD.
Yes, yeah. Now we move to the training case, the case study. So, as you know, quality by design can be applied to formulation development, to process development, to analytical procedure development, as we have seen. And with Thierry here, we have chosen a manufacturing process of a monoclonal antibody, and we'll present you the QbD aspect. So we will have four items. So now I move to the I remind you what is the manufacturing process of monoclonal antibody. So globally, the manufacturing process is divided into three main stages: the upstream process, the downstream process, and the fill and finish. The upstream process begins with the selection of a host cell for the production, in which you will be transfected the gene of interest, the gene of your antibody. Then this cell line will be developed. A cell bank will be established, and will be the beginning of this upstream process. There will be seed culture, expansion of viable cells, and then the batch, you have the fed batch, which lasts about 10 or 14 days, in which the culture will go, and in a bioreactor, increasing volume, fed with nutrients and raw materials. At the end of this upstream process, the harvest step occurs. The fluid containing the antibody undergoes a clarification step, and then we move to the downstream process with a series of chromatography systems to purify the antibody before the fill and finish of the drug substance or drug product into the primary packaging. In this case study, we will focus on the step fed batch with the different parameters to be studied in this step. Next slide, please.
Thank you, Nicole. So now it's time to start the application of Agile QbD to the fed batch unit. To start, we have listed on the right all the inputs. So we have listed 9 inputs, essentially material attributes and process parameters, and we have identified 4 outputs. Unfortunately, one, the map title or map yield, is not controlled as expected, in a sense that the acceptance criterion is not met. And so we have to solve that question. That will be the question all along the application. And what we propose is now to address that question, that is, can we improve the map yield, to decompose that question into 4 smaller questions. And for each question, we will use and apply a specific QbD sprint indicated below. So let's start by the first question. And this question is, what are a priori the critical inputs impacting the output, and the output being the map title? And so now to address that question, we have applied, indicated on the right, a small sprint, in a sense that only the first 2 steps, and particularly the IOM modeling, is implemented. And during this step, we apply a specific approach called FMECA for failure modes, effects, and criticality analysis, coming from the ICH-Q9 guideline. And in this approach, we compute a number, more exactly what we call the risk priority number. But this is a number able to assess the criticality of an input. But to compute that, we need 2 scores. And a score of severity given by a table on the left, and a score of uncertainty indicated on the right. In this situation, I have simplified the FMECA approach with only 2 scores. And so this score is chosen and selected by an expert based on the knowledge available. And for each input, the experts give a score about the severity. The severity here is the impact of an input variation on the output. So it could be an impact, it could be also regarded as a sensitivity. And so this is the first score, and score given for each input, but we need a second score given on the right. We need to assess the confidence level of the information source used to assess the severity. If the information is coming from a document published by the authorities, like regulatory agencies, the information is safe. But sometimes we have no information. If your technology is really breakthrough, disruptive, in that case, potentially we have no available information. So we are not confident, and in that case, this is a risk. So in that case, the score of uncertainty indicate why some inputs can become critical. And if we apply this approach based on the available knowledge on the fed batch unit, then we obtain on the next slide a specific diagram. This is a Pareto diagram. On the y-axis, you can recover the criticality score, and on the x-axis, you can see the 9 potential inputs. And this kind of diagram is easy to be interpreted in the sense that they classify all the inputs into 3 categories. In red, the critical inputs. On green, the non-critical inputs, and on the middle, the orange input corresponding with the potential critical inputs. So a first conclusion is, okay, we have identified 2 critical inputs, but we have a doubt, and potentially one of the 6 orange input could be in reality critical and could introduce some variability. So now that addresses a new question, and the question is indicated in this slide. The second question is, among the 6 potential critical inputs and on the real process, on the industrial process, what are the truly critical inputs? So this is the same kind of question. This is a question of criticality, but the first one was processed from the knowledge available. This now criticality question is based on data. So we can continue, and to do that, so now on the right, you can see we implement the complete sprint, and particularly the step number 3, the DOE, for design of experiments. People who have already implemented design of experiment know that there exist approximately between 5 and 6 categories of DOE. It depends on the question you want to address. And in that case, in terms of criticality analysis, we are using factor screening DOE. And more precisely, we have applied a plaquette-Burman design. And you can see on the left, the design of experiment. This is a table composed of 6 columns, each column corresponding to one tested input. And we have 12 different experimental conditions. We call also that a treatment. And each treatment was applied in triplicate. So finally, 36 assays will have been applied to the fed batch. I just mentioned that it is a simulation. So this is not real data, but virtual data. And when you have collected the data, you start the statistical analysis. And at the end of the statistical analysis, you can produce a diagram like this one. This is what we call a forest plot. And a forest plot is composed of 3 big columns. On the left, the column on the left is composed of the name of the 6 inputs and the different values taken by the input. And on the middle, you can see a graph. This is box plots, in fact. And on the right, you can see numerical values corresponding to estimation of effect, 95% confidence interval, and a p-value. If I go back to, for instance, if you look at U3, U3 is an input and can take in 2 values, 20%, 60%. And one of the 2 values, 20 in this case, is selected as a reference value. And this is why you can see a black square centered on the black dotted line. This dotted line corresponds to the null or the 0 effect. And when you switch from 20 to 60, you can see another box plot very far on the right from the reference value. So this difference, this gap, is really the effect of U3 on the mAb yield. And the effect is estimated to 2.69 grams per liter. So this is a big effect, and we can measure, we can quantify the effect. And if you analyze all the diagram, you can see that finally 2 inputs, U3 and U5, seems to get a critical effect. So finally, with this kind of diagram, we have a clear answer to the initial question. We have identified 2 additional inputs that could be, that are critical on the mAb yield. And now we can go to the next question. Since we have identified 4 critical inputs, the question becomes now, which range of values for the 4 critical inputs allow us to meet the specification on the mAb yield? And to address that question, we're going to implement the same kind of sprint, except that now initially we got 4 inputs, but one of them, U4, the feed solution, is a qualitative input, so it cannot be included in the study. So we fixed it, and we finally focus on the 3 remaining inputs, U1, U2, and U3, the feed rate, the dissolved oxygen percentage, and the agitation speed. And we selected another category of DOE, specifically to solve the optimization of response. And more precisely, we applied a Dollart design. So this is a design presented on the left on the table with 3 columns, and in this case, 13 treatments, except that we applied 5 times the operating condition at the center of the domain. So finally, 17 different experiments were carried out and applied to the fed batch. Like before, we collected the data and we analyzed the data in the last step of the sprint to produce finally a diagram like this one. This is what we call the probability diagram. Now, I just want to take 1 minute because in a lot of studies, people use a surface or response surface. But this kind of diagram, very popular, is not suited to this kind of problem because it does not take into account about the uncertainty. and uncertainty, as we've seen before, is really important. So finally, we prefer to compute this kind of diagram. And in the cube, this is a cube because we have to test 3 inputs, and each dot in the cube corresponds not to a predicted value of the MAB, but the probability to meet the requirement. So this is completely different. As you can see in this formula with the green frame, we estimate a probability that the MAB is greater than 5 grams per liter. If the probability at a specific point in the cube is greater than 95%, the dot is represented in green. If the probability is between 70 and 95%, the dot is represented in orange. And finally, in red, if the probability is lower than 70%. So the advantage is, first, we are taking into account the uncertainty. Once again, because the data are noisy, because we have a lot of disturbances, and it's easy to detect a promising region, potential region of interest to detect the future operating region. But before to go to the next question, it is important to inform that this diagram is based on predictions. So the next question is to test if the predictions are really efficient in practice. This is the objective of the last question. From the green region, from the promising region we have just identified, can we validate a specific operating region? And to do that, we once again, we repeat the same kind of sprint with a design of experiment, except that to simplify my speech and to make easier to understand, I have removed U3 to go simpler diagrams, but this is exactly the same reasoning. If you look at, we have 3 diagrams. On the left, you can recover the green region identified just before based on predictions, and we decided to test one specific operating region, more exactly a point, operating point. Why we say region? Because often it is what we call the normal operating region. When we say region, because if you, in practice, for instance, in a bioreactor, if you want to fix a pH, in practice, it's not possible to fix exactly the value of the pH. We have small variabilities. So this is a small region, but I prefer to say that in theory, we want to test one operating point. If you succeed to validate that point, you can go to the second diagram. In this second diagram, we wish to test one parameter, one input at a time. It means that, for instance, we fix U2 and we move U1. Or you can do the other, you fix U1 and you move U2 to test the possibility to get range in which the specification is still met. But I do not like really this kind of diagram because you do not validate a region, you validate, as you can see, across. The most really useful is the diagram on the right, because you try to, the ambition is to validate a region. This region is called the design space. This is the area represented in red, centered on the point we have validated during the first diagram. And to validate this, we test different points on the border of the square. And if you succeed to validate it, finally, you can extract from the zone 2 ranges, a range for U2 and a range for U1, and it corresponds to the established conditions. And if you can select any point within the region, for the authorities, it is equivalent. It is the same product. So this is why it's really important. And now I think it's time to sum up. And okay, at the beginning, we got a problem in the sense that during the production or at a small scale, we got a problem with the MAB yield. And 9 suspected inputs were identified. During the first sprint, we identified 2 critical from the available knowledge. During the second sprint, we used a design of experiments to identify 2 additional inputs. And during the sprint number 3, finally, from 3 critical input, we identified a promising region, but based on predictions. And finally, during the last sprint, we validate a sub-region called the design space to be able to extract the established conditions. So as you can see, this is an incremental approach to progress sprint by sprint and to see to visualize the progression of the development sprint by sprint. So now I think it's time to conclude, I suppose.
Yes, thank you, Thierry. So now we arrive at the conclusion and the takeaway messages we want to convey today. So first of all, QbD, as you can see, allows a close monitoring and robustness of the quality and manufacturing of the pharmaceutical compound. We have today we have a talk about pharmaceutical compound, but we can also say, I think, Thierry, that all those concepts can be applied to the development of medical devices as well.
Yes, and it's also important, as I said at the beginning, that we have developed Agile QbD to extend and, in fact, to democratize the QbD, to put this good practice of development to the preclinical phase of development, because for me, that probably is the most risky phase of development. And at this level, the Agile QbD can help to optimize the composition of the compound, but also to reduce the duration of the project. But my experience in practice is that if we want to successfully apply QbD, it's like a chemical reaction. We need 3 specific ingredients. We need the right methods with the right tools and the right experts working together to implement the QbD. This is the reason why at Cybernano we have developed Easy QbD. This is a web platform, and this is in fact a collaborative platform in which CMC expert, regulatory expert, biostatistician, developers can work together to collaborate and to correctly implement the QbD or Agile QbD.
And QbD is, as you have seen today, completely integrating the regulatory requirements and expectations from health authorities, EMA, FDA, Japan, and other countries as well, and are very much invested and will be more and more invested with implementation of the ICH M4Q second revision guideline that is planned to be adopted next year. This is the end of this presentation. Thank you for your attention.
Thank you, Nicole, Thierry, for your insights on QbD and more precisely Agile QbD. We've got a few questions received during the live session, but before we answer these questions, I would like to ask the audience one more and the last question. The poll question is, what's your primary objective with QbD over the next 6 months? It's an interesting mix of short-term and very pragmatic objectives. But what's important, and you can see from this presentation, is all these goals don't require a full-blown QbD program from the very beginning, from day one. The Agile QbD is really designed to be incremental, as Thierry presented, and scalable. All right, so now let's take a look at the questions we received. We have one, is how long can a QbD sprint last? And would it be possible to run several sprints at the same time in parallel? Maybe Thierry, you can share your comments on that.
Yes, in fact, that's a question often asked. And it really depends, as you probably remember, a sprint is composed of a step dedicated to the implementation of the experiments. When the experiments do not need, for instance, cells, it could be really quick. If you need in vitro experimentation, it's a little bit longer. And if you need to carry out in vivo experimentation on animals, it will be longer. So I could provide you between less of 1 week to several weeks according to the nature of experiments to be implemented. This is a kind of range I can provide you. And I think another question was to potentially to run sprint in parallel. That's
Yes.
also possible. Yes, to save time, a possibility is to address different questions in parallel by different sprints for different questions. It could be associated with a safety aspect, it could be associated with an efficacy problem, or it could be a problem of quality, a problem of associated with also the packaging aspect. It depends, in fact. And in that situation, we can clearly implement in parallel different sprints.
Thank you, Thierry. Nicole, you have been working on a lot of QbD related projects. What would be your recommendations on the CMC regulatory perspective?
Yes, yes. In fact, indeed, I have reviewed several dossiers or performed gap analysis, either at each step of stage of development, and I have some comments that clear, would say some common recommendations, because it's recurrent. I see sometimes that the clear list of CQA and CPP are not clearly defined in the dossier. The risk assessment are performed, but a clear list, the outcome of this risk analysis is not really clear, and should be derived from risk analysis and FMEA. And also a clear distinction between quality attribute and process parameters, or CQA and CPP, that has to be really defined. Also, what is missing often, I would say, it's the justification of the acceptance criteria and the ranges that are proposed for CQA, and especially for CPPs, for critical parameters. Sometimes we saw ranges, but we don't know where they come from. Also, we can see PAR and NOR, but normally the NOR should be included in the PAR, as we mentioned, we saw in the case study, but sometimes we have the same value. So I hope that today we have clearly helped to define what is a PAR, what is a NOR, and what are the different parameters concept. Also, sometimes I see some mix-up between CQA and critical in-process controls that are different, and even, or at least IPCs are release testing, release time, release testing. This of course can be a CQA at the end in the final product. But what is in process is in process, and CQA are on the final product. So this is really the main comments that I can make while reviewing all the dossier, including QbD.
Thank you, Nicole. That's very useful. I have another question, well, not me, but from the audience, is can the same Agile QbD approach be applied to analytical method development, ICH Q14?
Yes, I think I can answer. Yes, it's, for me, according to me, that's exactly the same approach when you have to, because it is more and more important, because we have seen that the development of a product and process based on QbD, we can regard this as a data-driven approach of development. So it's really important to get good quality of data. And in that case, if the different sensors, probes used offline, at-line, inline, do not provide robust and accurate information, that could be a problem for the QbD aspect. That's why the ICH Q14 is really important to not only develop the process, but to develop the analytical methods. And the approach we have seen for the Agile QbD can be completely applied to the development of analytical processes. This is the same kind of principle, not exactly the same aspect as reminded by Nicole during the first slide, because the acronyms are not the same. This is MODR instead of design space, and the notion of input, outputs are not process parameters, not material attributes, but other type of inputs. But behind that, the approach is strictly the same. So we can even imagine during the development to get sprints devoted as QbD for the product and other sprints devoted to problems of the analytical processes. That's completely possible.
Okay, I hope it's helpful with the answer Thierry provided. There's no additional questions from the audience. I think with that, we can close the session. Thank you very much all for your attention and engagement. We hope it's useful for your product development, and we are looking forward to seeing you again in VCR's next webinar.
Thank you.
Thank you. Bye-bye.
Thank you. Bye-bye.